let's come to the chapter 12. This is the most important chapter. So, biotechnology and its applications. So, coming to the biotechnology applications. So, previously we have learned what is biotechnology, principles of biotechnology, none other than the molecular genetic engineering and bioprocess engineering. And you have came to know the process. Coming to biotechnological applications in agriculture and biotechnological applications in medicine, transgenic animals and ethical issues. Let's talk about biotechnology. You would have learned from the previous chapter the essential deals with industrial scale. Production of biopharmaceuticals like production of insulin, production of any drugs, production of enzymes, and biological using of genetically modified microbes, fungi, plants and animals. Biologically we can modify these. The application of biotechnology includes therapeutics, diagnostics, means for the treatment, for diagnosing, genetically modified crops for agriculture, processed food, bioremediation, waste treatment. We already have seen in the secondary treatment, we mainly use this anaerobic digesters as well as aerobic. So, right, and energy production. So, three critical research areas the biotechnology are. So, okay, remember biopharmaceutical, genetically modification of microbes, which is bacteria, fungi, plantae and animalia. And next, genetically modified crops for agriculture, therapeutics, diagnostics, processed food, bioremediation, and we have the waste treatment and energy production, providing the best catalyst in the form of improved organism, including the microbe or pure enzyme, creating an optimal condition through engineering for the catalyst to act as. And next, downstreaming process technologies to purify the protein or the organic compound. So, we have to provide the best catalyst in the form of improved organism, usually a microbe or an enzyme, and optimal conditions for the catalyst to act. Downstreaming process technique to purify the protein or organic compound. Let us now learn how the human beings are used biotechnology to improve the quality of human life, especially in the field of food production and health. So, biotechnology application in agriculture. Let us take a look on three options that can be thought for increasing food production. Agrochemical based agriculture, organic agriculture, genetically engineered crop based agriculture. So, these are the three things in the biotechnological application. One is agrochemical based agriculture. We have to use chemicals. And next thing is organic agriculture. Next thing is genetically modified, engineered crop based agriculture. So, let's talk about this. Green revolution. This green revolution succeeded in tripling the food supply, but yet it was not enough to feed the growing human population. Increasing yields have been partly due to use of improved crop varieties, but mainly due to the use of better management practices. Use of agrochemicals, fertilizers and pesticides. However, for farmers in the developing world, agrochemicals are often too expensive, right? And further increasing in yield, existing varieties are not possible using conventional breeding. So, is there any alternate pathway that our understanding of genetics can show that farmers may obtain maximum yield from their field? Yes. Is there a way to minimize the use of fertilizers and chemicals so that their harmful effects on the environment are reduced? Use of genetically modified crops is a possible solution. Plants, bacteria, fungi, animals whose genes have been altered by manipulation. We call that as genetically modified organisms, GMO. So, genetically modified plants can be useful in many ways. genetical modification has made crops more tolerant to abiotic stress. They can able to tolerate more abiotic stress like cold, drought, salt, heat. So the increased efficiency of mineral usage by the plants. So actually what main thing is the plant has to tolerate the abiotic stress like cold, drought, salt, heat, and reduced reliance on chemical pesticides, means pest resistant crops, help to reduce post harvest losses, means it should not be rotten very early. Increased efficiency of mineral usage by the plants. This prevents early exhaustion of fertility of the soil. So enhance nutritional value of the food. For example, vitamin A enriched rice. Right? And mainly thing you remember, so climatic conditions like cold, drought, salt, heat, these things should be tolerated. And reduced reliance on chemical pesticides, means pest resistant crops should be developed, and post harvest loss has to be minimized, and to decrease the fertility of the soil, so usage of the minerals has by the plants. Nutritional value also has to be developed. In addition to these uses, genetically modified has been used to create tailor-made plants to supply alternative resources to industries. So in the form of starches, fuel and pharmaceuticals. So some of the application of biotechnology in agriculture that you will study in detail are the production of pest resistant crops, so which could decrease the amount of pesticide usage. For example, Bt toxin is produced by bacterium called Bacillus thuringiensis, Bt for short. Bt toxin gene has been cloned from bacteria. So Bt toxin, Bt is the gene they extracted from bacteria and has been expressed in plants to provide resistance to insect without need for insecticides. In effect, created a biopesticide. For example, we have Bt cotton, Bt corn, Bt rice, Bt tomato, Bt potato, cotton, corn, tomato, potato, rice and soybean. So these are the varieties that is been produced by Bt. And coming to the very first thing, Bt cotton, how it works. So Bt cotton, some strains of Bacillus thuringiensis provides protein that kills certain insects such as lepidopterans. So usually we call it as tobacco budworm or armyworm. So we have tobacco budworm or armyworm. Coleopterans, diphterans. So some strains of Bacillus thuringiensis will produce a protein which can able to kill lepidopterans, coleopterans and diphterans. Okay, none other than tobacco budworm and armyworm. So coleopterans is beetle, diphterans is none other than flies and mosquito. So Bacillus thuringiensis forms a protein crystal during the particular phase of their growth. So these crystals contain a toxin which is insecticidal. Why does this toxin not kill the bacillus? So actually Bt toxin protein exists as an inactive prototoxin, but once an insect ingests the inactive toxin, it is converted to an active form of toxin due to alkaline pH of the gut, which solubilize the crystals. The activated toxin bind to the surface of mid-gut epithelial cells and create a pores and causes the cell swelling and lysis and eventually causes the death of insect. The specific Bt toxin gene are isolated from Bacillus thuringiensis. Okay, the specific Bt toxin gene are isolated from Bacillus thuringiensis and incorporated into several crops. We already have seen, right? Bt cotton, Bt corn, Bt soybean, tomato, potato, rice. So isolated in the and incorporated into several crops such as cotton. So this is the picture showing you the cotton ball destroyed by bollworms. So this is fully matured cotton ball. So this is destroyed by the bollworms. So the choice of genes depend upon the crop and the targeted pest. As most Bt toxins are the insect group specific, the toxin is coded by the gene name called Cry. So there are number of them. For example, the protein encoded by the gene Cry1AC and Cry2AB control the cotton bollworms. Okay, so remember Cry1AC, Cry2AB. This will control cotton bollworms, and Cry1AB controls corn borer. One AC and 2AB, and Cry1AB controls corn borer. Okay, this we have to remember. Next thing is pest resistant plants. So what is this is? Several nematode parasites, a wide variety of plants and animals including human beings. A nematode, Melody incognitia, infects the roots of tobacco plant and causes great reduction in the yield. A novel strategy was adapted to prevent this infestation, which was based on the process called RNA interference. RNA interference takes place in all eukaryotic organisms as a method of cellular defense. This method involving silencing of the specific RNA due to complementary double-stranded RNA. Molecule that binds to and prevents the translation of mRNA, that is none other than the silencing. The source of this complementary RNA could be form of an infection by the virus, having RNA genomes or mobile genetic elements. We call it as transpoons that replicate via an RNA intermediate. So here RNA could be an infection. So this complementary could be an infection by virus having RNA genomes or mobile genetic elements. We call that as transpoons. Replicate via an RNA intermediate. So, using the agrobacterium vectors, nematode specific genes were introduced into host plants. So, we all know agrobacterium tumefaciens, right, which will produce the tumor. So, with that, nematode specific genes were introduced into host plant. The introduction of DNA was such that will produce both sense and antisense RNA in the host. Cells, these two RNAs being complementary to each other form a double-stranded RNA that initiated RNAi and thus silenced the specific mRNA. So, of the nematode. So, the consequence of that parasite could not survive in transgenic host, expressing the specific interfering RNA. So, the transgenic plants therefore got itself protected from the parasite. So, here you can see host plant generated the double-stranded RNA, triggers the protection against nematode infestation. The roots of typically control plants. Typical of control plants. This is A. And this is the transgenic plant root 5 days after deliberate infection of nematode, but protected through novel mechanism. So these are the things we have to remember when it comes to this topic, right?